Okay guys, today we're going to talk about cellular respiration, and it's just an introduction. So you're going to hear a lot of terminology, and I want you to know that this lesson overviews, or is an overview of three or four different lessons that we're going to go into more detail. So we're going to just briefly talk about each one. So a couple things to remember from previous lessons, even going back to grade 9. All energy comes from the sun, and this is called radiant energy. And we know that plants use this energy to make their own food, glucose specifically, through photosynthesis. Nicely, oxygen is a byproduct, and the plants need carbon dioxide and water. Again, which is nice, taking the carbon dioxide out of the environment. All we have to do is water them. Okay, also, our cells, or also plant cells, so it's important to remember that cellular respiration occurs in plants and animal cells. They metabolize glucose, our cells metabolize glucose, and this is through a controlled oxidation process. So the lesson that we don't do in the book talks about how you could break down glucose really quickly, but a lot of the energy would be released as thermal energy, and it wouldn't be able to be used to drive other processes. So it wouldn't be able to make ATP. So instead, there's a whole bunch of reactions that occur slowly that are able to then trap the energy and make it into usable energy. Okay, this slide is busy. So we're going to just go over everything I'm saying here, and then we're going to look at the diagram together. So one definition I want you to highlight and make sure you have this is aerobic cellular respiration. So I don't know how much you can see. You might be like, why doesn't this look sweaty and red in the face? I'm recording this on purpose right after I've run on the treadmill this morning. So yeah, it is 4 a.m. Aerobic cellular respiration is using oxygen in order to essentially break down glucose and get the energy from the glucose. So the important thing to remember, aerobic is using oxygen in the reaction. And again, we're going to go into all of the details. That's going to be in contrast to another term that we're going to look at in a second. So remember, products of photosynthesis are used, i.e. glucose, are used to release energy for the cells. This is done to produce ATP, and we did a whole lesson on ATP, so no need to review that. And again, we remember that the chemical energy is stored in that last bond that attaches the phosphate to the ATP. And once that bond is broken, that energy is easily released, which can then be used to drive another reaction. And that's the whole purpose of having the storage of ATP in a cell. When oxygen isn't available, cells use anaerobic cellular respiration. Okay, and we're going to look at that in just a second. What I want you to look at first is this diagram is just from grade 9. It's just showing essentially how photosynthesis and aerobic cellular respiration are opposing reactions. So we have water and carbon dioxide go into the plants for photosynthesis. Sunlight is the driving force that allows photosynthesis to happen. And oxygen is a beautiful byproduct, and this is done to produce sugars, which have a lot of energy in the bonds. Sugars then are broken down in both plant and animal cells, needing oxygen. Okay, and again, this is aerobic cellular respiration. ATP is produced, and water is a byproduct, as well as carbon dioxide. Okay, so aerobic cellular respiration takes place in most eukaryotes and some prokaryotes. Okay, remember these terms from grade 11. Most eukaryotes are obligate aerobes. This means that they cannot live without oxygen. So break up the words. Obligate, they're like obligated, maybe, to use oxygen for cellular respiration. So, for example, humans, we cannot live without oxygen. Aerobic cellular respiration, again, we're extracting energy from the glucose and we need oxygen. So it's just emphasizing the fact that aerobic cellular respiration demands the use of oxygen. And again, ATP is synthesized, and we remember the terms ADP and inorganic phosphate. And this is from the cytosol. ATP, again, can be used to drive other cellular reactions. Okay, this diagram is right from your textbook. It is an overview of what we're going to be looking at. So it might look like it's simplified, but it might look like a lot is going on. And that's A, because it's true, but B, recall that we are going to be looking at each stage by itself. Okay, so the first stage is called glycolysis. This is the only stage that occurs in the cytosol for all cell types, whether they're prokaryotic or eukaryotic. Maybe you can tell me why with prokaryotes it's definitely in the cytosol. Hopefully somebody can tell me that. And then all of the rest of the reactions occur in the mitochondria with eukaryotic organisms. So the second stage is pyruvate oxidation. And again, we're going to talk about each of these as we go. And we have citric acid cycle. And then lastly, we have an electron transport chain. And so this is what I call follow the electron, which we will do. Looking at each stage, we have ATP produced here, here, and here. But notice that in this stage, it's called substrate level phosphorylation. So the definition is here, and make sure you have this highlighted. So this is where ATP is directly made from some sort of reaction catalyzed by enzymes. Okay, so it's directly made. You'll get a better sense of this as we go through these types of lessons. From your experience, probably, this is the only type of ATP production that you're probably would be familiar with. So this is kind of the cycle that we go backwards in the video. So this is like the cycle that we drew on the whiteboards the other day. Okay, oxidative phosphorylation is when ATP is indirectly made through a series of reactions. And again, this is with the electron acceptor at the end, which would be oxygen for aerobic cellular respiration. Okay, so let's just talk about the stages briefly again, just so you have just a baby idea of what we're talking about when we talk about each reaction. So again, the first one is glycolysis, and there's a lot of reactions that happen, and we're going to look at this with each of these with various activities to make it a little more palatable, and it'll actually internalize a lot easier than just making you sit and memorize. So glycolysis, again, occurring in the cytosol. It's the only one that occurs in the cytosol in eukaryotic organisms. It is a series of reactions in which the glucose molecule, so the starting molecule is glucose. That is your clue for tomorrow. I'm not telling you anything with your activity, so take all the clues you can. And it gets broken down into two pyruvate molecules. And you'll get familiar with what all these intermediates look like and the enzyme reactions. We see that ATP is produced. In this case, in glycolysis, only two net ATP molecules are produced. Pyruvate oxidation is the shortest reaction, and this is where pyruvate is oxidized by NAD+. So what is NAD+? NAD+ is an electron acceptor. So it can be reduced to NADH, and it's going to carry those electrons to the final stage, the electron transport chain. So it's literally called an electron carrier. We see there's another one down here, FADH2 is another electron carrier. So it's just a means to get electrons from one molecule to where it needs to use those electrons to make energy. Okay, again with pyruvate oxidation, we have the NADH being produced, and carbon dioxide is also being produced. Okay, so with the citric acid cycle, it's a cycle, so it's circular, and you're going to see that in activity as well. And again, we have a series of reactions. We have ATP produced, so directly we'll have ATP produced, and we will have NADH and FADH2 produced. And again, those are electron carriers that are going to take electrons to the final stage, the electron transport chain. And then again, we will talk about all of the reactions and steps and the importance of getting electrons to the electron transport chain. Okay, this is an overview of all of the reactions, or I guess stages. Again, glycolysis, we have glucose producing 2 pyruvate molecules and 2 ATP. Then that gets carried into the mitochondria and all the fun begins. So again, the pyruvate oxidation is very quick. It's a short cycle. We have the Krebs cycle, which is the same thing as a citric acid cycle. So if you talk to an old person, we call it the Krebs cycle. The name was changed to citric acid cycle. Same thing. The terms are used interchangeably. And here we have 2 ATP produced, and we have, again, a lot of NADH and FADH2 produced, which are going to carry electrons to the electron transport chain here, where we get a boatload of ATP produced. And this is where all of the electrons that get carried to it are able to help create the ATP. So the ATP is created differently in this situation than it is in the Krebs cycle and glycolysis. So the mitochondria shouldn't be new at this point from grade 8, grade 10, and unit 1. So we're not going to go over it too much, but remember the glycolysis occurs in the cytosol, but everything else occurs in the mitochondria. The mitochondria is considered the powerhouse because this is where energy is made. And recall that we have 2 membranes, and this is really important just to start to get familiar with the structure of the mitochondria. We have the outer membrane, and we have this inner membrane that creates folds. So we have the intermembrane space. So inter is between. So intermembrane means between the membranes. So we have the space between the membranes. So that's the intermembrane space. We also have the matrix. So we'll get to get into the matrix. And this is the interior aqueous environment of the organelle. Both the matrix and the intermembrane space are really important for all of those processes. So it's important for you to get your mind wrapped around what is what, because we will talk about things happening in the matrix and then things being exported to the intermembrane space. You want to have that visual in your mind so you understand what's happening. Okay, prokaryotes. You're thinking bacteria, that's where your brain should be going. This is what I studied. So all these organelles, when we're talking about them, are just like luxury items, in my opinion, because prokaryotes don't have that compartmentalization. So with prokaryotes, some will undergo aerobic cellular respiration, even without the mitochondria. And that's because they don't have any. So that answers my question from earlier. So all the processes will occur in the cytosol, and we can see here, and we see the pictures. This is a good picture here. So we have these extra folds, and it actually kind of looks like a mitochondria, and they're just extensions or derivatives of the plasma membrane, where the electron transport chain or the other processes can actually still occur. And you'll see again how cool that is when we actually learn about each process separately. So this should not look familiar to you. This is the electron transport chain. So this is one we haven't looked at yet in the other diagrams. And we start on the left. Where's my mouse? And we watch in the red, these are the electrons. And they're moving from different proteins, different enzymes, different complexes until they get to the strongest oxygen at the end. And in doing that, it is able to produce ATP. We'll talk about that. Okay, anaerobic pathways. The whole reason why I'm now cooled down. But originally I wanted to do this on the treadmill, and I realized I'd probably break my laptop if I did that. It would fall. So anaerobic pathways, these are getting energy from glucose or foods without oxygen. So if I'm just jogging, and being able to talk, so that's the jog test. If you're at a comfortable jog, you can have a conversation. That's aerobic. There's enough oxygen getting in you that you can maintain that pace forever. So that's essentially what I'm doing for the most part when I'm running really long distances. So that's aerobic. Anaerobic is when you are running so fast that you're breathing while you're running, but then once you slow down, or you have to slow down at some point because your body just can't keep up. And once you slow down, you are gasping for air. You might have that pain here, which I started to get on my run, and where it feels hot and burning, and that's great to have. But in those scenarios, you're switching to anaerobic respiration. There's not enough oxygen to support the making energy that you need. And again, we'll talk about this briefly today, but more so later. Okay, so anaerobic, you can remember anti-aerobic, antioxygen. Fermentation, we might be familiar with some fermented foods that we've heard of. If you can tell me any, I'm not going to tell you what they are. Supposedly really great for your digestive health, really good for your gut microbiome to be consuming fermented foods that haven't been processed. So you don't want them heat killed. So they need to be live, I think is what they call them. So fermentation does not use an electron transport system. So that last one at the end where the electron is passed from enzyme complex to enzyme complex to oxygen. It relies on an organic compound to act as the final oxidizing agent. So it's very different than what we're going to study at first. So a common reaction is fermentation of glucose, where ethanol is produced in carbon dioxide. What I want you to notice is the Gibbs free energy being negative 218. Go back and compare that for what the Gibbs free energy is for the oxidation of glucose. Okay, so go back and compare and tell me what process seems to be better. Okay, and examples of microorganisms that use the various forms of respiration that we talked about. So we have Clostridium tetani and Clostridium botulinum. So botulinum, so both of those are bad for you. What's the word? Produce toxins that can kill you. So botulinum is the botulism bacteria. So probably not an issue these days, but It used to be a problem with like eating food in old tin cans, so now they've been changed to a different material. They now have lining that prevents any kind of like food. The food can keep for a long time, and typically it's the bacteria. There's no bacterial growth. You can probably some really old canned food. But what's interesting about that bacteria is the toxin that causes botulism to be toxic is what they inject into maybe your face for removing wrinkles. If you've looked closely enough at me, I don't get that done. My friend does, and I think you can tell, in my opinion. So those are obligate anaerobes. So these are organisms that cannot live with oxygen. So let's think of the environment of living in a tin can. They have no oxygen for the botulism one. Okay, so they cannot survive in the presence of oxygen. They use, so this is just kind of like a little introduction. Again, we'll have more of a lesson on that. They use typically sulfur or iron as their final electron acceptors. And we'll talk about that more later. I want to start telling you more, but last slide. And then there's other organisms that can live without oxygen, but they can also live with oxygen, and those are called facultative anaerobes. So they can survive with oxygen. They do better without, but they can live with both. So probably optimal. And E. coli we're familiar with. We have E. coli in our systems all the time, but it's when we get an imbalance of E. coli that is what makes us sick. And this is often from beef, I believe, contamination. And if you want to know why, just watch a video about how beef is produced and you'll get an idea. Listeria is another one, and that's from pork. Tends to be one. I don't think it's too common anymore. And then Staphylococcus epidermis is like the staph infection, and I point at my arm because that tends to be like you get that kind of skin infection that can be dangerous. Okay, so that is it for the overview of this whole lesson. I know there is a lot of information, but remember we are going to talk about each one separately. So we spend 2 days on glycolysis, and then we'll spend a day on pyruvate oxidation because it's really quick. 2 days on the citric acid cycle, 2 days on electron transport chain, and then we talk about anaerobic cellular respiration, where we get to talk about some of these other interesting things. So it is stretched out. So it's just good to have an idea of what happens after glycolysis. Now you know that like, okay, pyruvate's made, big deal, but now you know like, okay, so it's gonna go here and then here. So you have an idea of the end result. Okay, so make sure your notes are filled out, and tomorrow we are going to tackle and address glycolysis. Have a good day, guys.